Good evening, everyone. I'm Professor Jason Adolph in the biology department, MEVP program at Monmouth University and the Urban Coast Institute. And I want to welcome you all here and thank you. I think you'll be thanked many times over the course of tonight for participating in these programs and helping us out with what we're trying to do. My job here tonight is to give you a background on what it is that we do and to, because it's a little different than normal fishing. And to introduce Rich, who is going to tell you about the results of the data that he collected. But first, there's people in this lab who are not going to be up here presenting tonight that I want to acknowledge. The way I think of this story, when Rich got hired into my lab here at Monmouth, we needed someone who could do both field work on boats, shorelines, the dirty, wet stuff, and laboratory work with molecular biology at the bench. If you know Rich has the former part nailed, and he had some experience with molecular biology. And I said, Rich, can you cook? Do you cook? And Rich said, yeah. I said, okay, you'll be able to learn molecular biology, no problem. But my lab here includes these two people. Eric Conlon was a Monmouth student who's been in my lab since 2018 on different projects, was a student here, graduated, has worked full-time, is one of the more consistent laboratory people that I've ever worked with. And Liz Clark joined the lab in January of 2025, I believe it was, with a master's degree doing molecular biology and molecular assays of sea creatures, sea turtles in particular. I'll take like 2% credit, and the rest goes to Liz and Aaron in helping Rich become the laboratory molecular biologist that he is today. Be able to do this stuff. And that is how science works. If you're a scientist, you're always learning and you're always teaching. So in our lab, we work with something called environmental DNA. We work with fish in this context. We want to know about fish in the ocean, in the rivers, in the estuaries, in the lakes, whatever. But we don't fish. No fish were harmed in the production of this data. Some think that's the most boring way to learn about fish, and we think it's a cool way to learn about fish. We don't touch fish very often unless we're working on someone else's project. We work with stuff called environmental DNA or eDNA that you hear about. And it's the DNA that's in the water that's separate from the organism that produced it. And it's like thinking of forensic science in a crime scene. When a forensic scientist goes into a crime scene and starts swabbing things to find DNA that was left behind by the people that were there. That's what we're doing. We're finding traces left behind of the fish that are in the ocean instead of capturing the fish. And that's a really efficient way to learn about fish. So efficient that our federal government and European governments have kind of codified it in national strategies to start using eDNA in conservation biology and conservation management, because it's so much more efficient for some things than actually capture surveys, capturing fish or capturing invertebrates and stuff like that. That was a Biden White House thing. I hadn't heard much of it since. So the rundown of what we do. So imagine a fish in the water, okay? Fish sheds scales, mucus, cells, feces, urine, all sorts of stuff. And in those materials, there's DNA. Extra credit, what does DNA stand for? Anyone?
Deoxynucleic acid.
So close.
Yes. That's ribonucleic acid.
Yes, yes, you got it. So this is the stuff that's in every organism's cells in our genome. And it's written out in like letters. They're chemical compounds. We can think of as letters A, T, C's, and G's. And it's the code of life. We share some sequences with everything, many things, and some of the sequences are unique. So when a fish sheds its DNA into the water, it becomes environmental DNA. It lasts not very long in the water. You can detect it for 3 or 4 days afterwards. So if we find it, we know the thing was there recently. This is what you all did. You went out with bottles and you collected water and brought it back here. Those bottles of water contain microscopic bits of DNA, environmental DNA, that then Rich analyzed in the lab and got the results that he's going to show you today. And the reason this works is because once you have the isolated DNA from the environment, you can pick out a gene from the genome using something called primers. And it's just to say of the millions and cajillions of base pairs in the DNA in the water, we're only interested in a 109 base pair, 109 letter long word that is unique to a type of fish. So we use primers to find that and make lots of copies of it from the DNA that you collected. Then we sequence that DNA, literally read the letters, and then match that up to a reference database. And we say, oh, that word means sea bass, striped bass. That word means black sea bass. That word means American eel. And we can count how many different types of fish DNA were in that bottles that you gave us using those sequences. I just simplified a really, really complicated process. But that's basically it. You bring us DNA, we sequence it, tens of millions of DNA sequences where we deal with a data set like this. We figure out what words are there and how those relate to different fish. So obviously somebody has taken a striped bass, taken a fin clip, gotten the DNA and figured out what sequence is unique to them. And that's happened for all these fish that you'll see up here tonight. So it's a really efficient and useful way to determine the community of fish in an environment. This is a graphic that I always show that's like what we found in one of our projects that we do in the state of New Jersey offshore the continental shelf. So we've detected 26,000 ASVs. The ASV is basically a fish gene. 60% of those are teleosts, or the bony fish, the common fish. And if you look and you know anything about the fish in New Jersey, these are the kind of things that we find. And it's not surprising. 10% of the sequences are the elasmobranchs. These are the sharks and rays, cartilaginous fishes. And a lot of them, again, are common. Smooth dogfish and spiny dogfish and these rays are very common. Thresher sharks, people pull them up on the shoreline every once in a while. There's a big fishery for them along the coast. This was a surprise to us. The basking shark. How many people didn't know there were basking sharks living off our shores? Basking sharks don't have teeth, not a big threat, but they're really cool. And of course, jaws. We found some jaws DNA. But because the primers that we use are common to all vertebrates, we also find marine mammals in the water. And this is the list of types of marine mammals that we found, and other non-marine mammals, sorry, Liz, and birds. Using this same, dolphins are all over the place. We have lots of humpback whales at times off our shores. The northern right whale, of course, is a very threatened, endangered whale species. And guess what? Not Neil deGrasse Tyson's DNA specifically, but human DNA is also all over the environment. And we find that, but it's a type of human DNA that is, you can't use to distinguish people, but it can tell a person from something else. As common as normal, it's all over our environment. the closer to shore and the closer to Manhattan we sample, the more human DNA we find. And then we work hard, especially our colleague Shannon O'Leary up in New Hampshire, to bring this number as close to zero as possible. The DNA sequences that we find in the ocean that we don't know who they belong to. Human, bird, mouse, deer, rat, fish, shark. We have no match for it. And that's something we're still working on. So the last point I want to make about this, and this is something we work on at the lab, and that Liz is leading a publication on. In order to detect all these things the traditional way, you would need an armada of boats and traps and trawls and nets. And we can find all this in the bottles of water that you're sampling. That's the efficiency of this method for the purpose of biodiversity. So this was my first attempt at making an AI-generated drawing. I'm sorry. Horrible. But the promise of eDNA meta barcoding, which is this is referred to, is the promise of going to the supermarket and just, you don't have to know what you're buying, you just drag it across the thing and it beeps and it knows it's a turkey sandwich or a can of sauce or a can of beans or whatever. It's got a barcode. That's where people like us are trying to go with fisheries science and conservation science. Can we, apply those barcodes to nature and assess biodiversity that way? The other thing is that things like you all have done here for this project would not be possible if I asked you to go out and trawl for fish or even seine for fish to get an idea of the fish biodiversity around Monmouth County. That's, you could do that, but this is much more scalable the way we do this. We can actually collect bottles of water and bring them back to us. So we're trying to pilot that and develop that program. So again, thank you for your participation. Okay. So now it's my pleasure. I've already introduced Rich a little bit. We all know he's an excellent chef. Yeah. Right? Rich graduated Monmouth. I don't remember which year. That's my fault though. 3 years ago or so. 23? Yeah. Left us, worked for Rutgers, Haskins Shellfish Lab, worked on boats, doing clam surveys, and came back here for this project that's funded through Congressman Pallone and the NOAA Northeast Fisheries Science Center, looking at using eDNA to look at the biodiversity around Monmouth County. So I'm going to turn it over to you, Rich.
Excellent, thank you. So, hi everyone. I want to thank Jason for the introduction. I want to thank the rest of my lab mates for helping me out with this lab, some of this lab work, and just providing a excellent culture in which this work can be done. So I'm excited to get started with our community science presentation for the summer of 2026 results. So let's get going. As we begin, the first thing I want to talk about is kind of just some critical concepts of community ecology. Specifically, we're going to talk about biodiversity and community composition. So what biodiversity is it refers to the abundance of all life on Earth, the abundance and variety of all life on Earth. So far, we believe we've described about 220,000 marine species. Of that, 16,475 are marine fishes, and their estimates vary, but we may have as many as 428,000 to 741,000 marine species left undescribed. So of this tremendous variety of all life on Earth, what's amazing is that every single one of them, each unique species, contributes in some unique way to the functioning of its ecosystem. And as we lose species, or as they're reduced to levels that are no longer ecologically relevant, the ecosystem loses that function. And slowly the function of a whole ecosystem can be gradually kind of eroded away. So biodiversity is really something critically important. It's really something that we should be caring about. This kind of raises the question, how do we measure diversity? Well, we have a like nice painting of a coral reef up here, and we're going to kind of walk through an exercise of how we measure biodiversity. So there are two ways. Is there species richness, and that kind of just refers to going through and figuring out how many unique organisms are out there. And then we can use the actual counts of how many organisms there are to calculate a bunch of diversity indexes. We're not really going to be talking about diversity indexes today. Those will be in the results that I share with you in the future, so keep an eye out. I'll leave this in the slides just so you guys can kind of learn about that a little bit more. But in addition to community composition and the actual sheer numbers of animals in an environment, we also care about what animals are present in that ecosystem and what their proportions are. And the reason we care about that is because they have really significant implications for the ecology of an actual environment. Most famously, what you guys would probably have heard about is the case of the reintroduction of wolves into Yellowstone. It's a long, complicated story, but is the short version of it is basically there once were wolves in Yellowstone. We hunted them basically to a local extinction. They were no longer ecologically relevant. The elk population increased. They went through, ate all the vegetation. This changed the entire character of the river valleys of Yellowstone. Once wolves were added back into Yellowstone, kind of everything reversed itself and started to basically to mend and revert back to the state that it was once. So it kind of went back to how it used to be. The marine and estuarine ecosystem, similar things can happen is despite being in the ocean, it's kind of out of sight, out of mind, but we can feel the ecosystems, they suffer anthropogenic impacts, just like terrestrial ecosystems. And this is particularly true in the where we live in the lower Hudson River estuary. We do a lot of things. We urbanize the watershed, so we build a lot of stuff. These things like parking lots, increase impenetrable surface. We build dams up in the Raritan River, stuff like that. We also pollute tremendously, and this has implications on community composition. One example, probably the most egregious example of marine fishes and the reduction of marine fish is the river heron. So we used to have a lot of river herring. They used to come and spawn up in the Hudson River. They used to spawn in the Raritan River, and they even used to spawn in the Shrewsbury and Navesink rivers. There were a ton of them. They were incredibly prolific. But as we built dams, as we polluted, the proportion, they declined to only a shadow of their former self. And we can see, so this is from a study in the Gulf of Maine. It's a food web, so it kind of tracks the flow of energy from these primary producers and from detritus through the food web up to the river herons and then finally to the apex predator. And we can see that even in their diminished state, that the river herons are an incredibly important ecosystem. So we can imagine the implications for, having what used to be a really large proportion of animals suddenly decline to only about that much. So it's a healthy ecosystem. Ultimately, we need to conclude that a healthy ecosystem has a diverse community of species that across a wide variety of niches, and they're proportioned in such a way as to be trophically productive. So energy flows through this system naturally. So we've taken some time to discuss kind of the very basics of community ecology. And now we're going to talk about our preliminary results. We still have a little bit of work, so some of this might change, but this is basically what you guys are going to be sent. So here we have our summer 2026 science sites. We've sampled 24 stations. over the entire Hudson River and estuary, and then our additional estuaries as well. We got 9 sites in the ocean, and then 15 sites up here in the estuaries. And we did a great job. Thank you guys very much. Give yourself a round of applause. Couldn't have been done without you guys. So we did more samples in less time than the last sample got. So this has been really good work. It's been phenomenal. So now we're actually getting into the actual results. And what we see is we found a species richness of 79 organisms. We believe there are different ways to calculate and estimate species richness. Our estimates are between 87 and 104. Our best estimate, the Chow estimate, is going to be 97. So with that being said, we've accounted for roughly 81% of total richness in this environment. In addition to making these estimates, we're able to make something called a species accumulation curve. So what we do is we basically, for each one of these lines along the x-axis here, we're basically going to say, okay, n is equal to 1. We'll take one sample and count the unique species richness. And we'll do that. So we'll do that for n of 1. We'll do that for n of 2, and et cetera, all the way down the line. And what we get is we get this curve that we're able to fit a line of best fit to, and we get an equation. And with that, we're able to figure out, okay, we can go through and figure out how many more samples, how much more work do we have to do to find more and more species. So what we're able to say is that to find 95% of all the species present in our waters, we would need an additional 47 more samples. So we basically need to triple the effort that we did. But the other interesting piece of insight that we get from a species accumulation curve is we learn that we collect most of the species very early on. So we found 50% of the species within 4 samples. So 1, 2, 3, 4. So right around here, we found 50% of the species that are present in our waters, which is pretty cool. Now we're going to talk about species richness at each actual site. So the first thing we're going to look at is we're going to be looking at this map. Up here, we have the ocean sites here in these purple squares. And we kind of see a little bit of a fluctuating, like this fluctuating pattern of species richness. So up in Sandy Hook, we have the highest or fairly high richness, 34. In South Sandy Hook, 15. Seabright, 24. Out in Long Branch, we have a species richness of 39. This was the highest ocean richness. Further south, we have 23, 30. You can barely see this purple dot here, but there is a station here, and we only had 2 species represented at this site. That seemed odd, and it is odd. This station didn't have particularly good sequencing results. So with better sequencing results, we're gonna get higher richness, basically. So we don't trust this result, but we'll investigate further. Further on south, we have 29, and then at the mouth of the Manasquan River, a station of 23. So now we'll consider the estuaries, and we're looking at these orange dots here. So in the Manasquan River, we kind of have some moderate to high richness. 21, kind of a moderate level of richness. Then 26, fairly high richness. Up in the Shark River, we have really quite high richness. We have a site with 24 species present. And then here at McCleary Park, a site with 38 different species, which is really quite high. Up in the Shrewsbury and Navesink, it's a bit of a mixed bag. We have some sites that are fairly low, 14 here in the Swimming River, 14, 11, 17 up in Navesink. But we also have some sites that are more, more closely aligned to the median. So we have 20 species present in Manhasset Creek, and then 20 species present up here in the Shrewsbury Narrows. So as we are moving up into the Bay Shore, we kind of see it's a bit of a mixed bag again. Here we have 20, which is right around the average for the estuarine sites. But we also see 18 and 17. We also have 31 and 42 though. So like I said, a bit of a mixed bag, probably dependent on tide and habitat immediately surrounding these stations. So we've talked about geographically how richness is distributed. Now we're going to talk about how they're distributed like abstractly. So what we're looking at here is these are called box plots. And what a box plot wants to show us is kind of how the data is spread out. So we can see the spread of data. Each black dot represents a data point. This red dot represents the mean, so like the average. And this black line represents the median. For what we're talking about today, we're concerned with the median. So we can see that we have a median richness in the estuary of 20 species. And in the ocean, we can see this black line, trace it back over, and we have a median richness of 24. Now, it's kind of, we're not very good at doing this, is humans, humans that is. We're not very good at making that kind of a distinction. But what we do is we use something called a statistical test called a Wilcoxon rank sum test. And we're able to do the math, and we're able to look and figure that we did not find a difference in ocean richness and estuarine richness. So despite the fact that the ocean was higher, the gap by which it was higher, we couldn't say for certain that that's actually true. So this is what we're left with. We have to say there is no difference between ocean richness and estuarine richness. So, but this is the second sample bout though, and we get to do something I think that's really fun, is we get to compare the results from our summer sampling bout to our fall sampling bout. So here we have, again, series of box plots, estuaries out here to the right, and to the left, we have our ocean sites. Our summer sites are in blue, and our fall sites are here in red. So the first thing that I notice is that the median richness between the summer estuarine sites and the median richness between the in the ocean fall sites are very similar. That's interesting. We'll touch on that later. So keep that in the back of your mind. The next question I ask when I'm looking at this is there a difference between the median richness from the fall and the median richness from this recent sampling bout? And when we do the math, we find, yes, there is. We find a Wilcoxon rank sum test. It yields a p-value less than 0.001. So there is a statistically significant difference in richness between sampling bouts in the estuaries. So we'll give the same treatment to the ocean sites. And the first thing you'll notice is the gap is much smaller. So that kind of hints us off, that should give us an idea of where this is going. When we do the math, we find a p-value greater than 0.05. So we have to say there is not a difference between ocean richness in the fall and ocean richness in the summer. So as we said, richness differed in the estuaries, but not in the ocean. So what happened? It's kind of an interesting result. Why did we have richness? Why was there a difference in richness in the fall between the ocean and the estuaries, but not a difference in richness between the ocean and estuaries now in the summer? So basically what happened is that the gap between mean estuary richness and mean ocean richness, it shrunk. And the vast majority of why that happened was because the estuarine richness was increasing at a faster rate than the ocean increased. So basically, what's happening is that we believe that there's seasonal migrants from the marine environment coming into the estuarine environment. This is a well-documented phenomenon in our area. is one paper says that 82.5% of marine fish use estuaries at some point during their life. So we know that these are incredibly important habitat, and we know that fish come at different points, they come and use this habitat, and that's almost certainly what we're seeing here. So the difference in richness decreased because of migrants, marine migrants into the estuarine environment. So we've discussed species richness, and we're kind of going to put that behind us. What we're looking at now is community composition. So this is basically what we have here, donut plots, and it represents the fish and what their proportion is. And you can see that graphically here as well. So we'll start in the ocean, and we see that we have Atlantic or Gulf menhaden, and then Atlantic menhaden or river herring. For our sake, we're going to treat these collectively, but they represent 24.8%. They're an incredibly important forage fish in our area. They're incredibly prolific. There are a lot of them. Books have been written about them. They've been called the most important fish in the sea. They're effectively the base of they're the primary consumer of our marine food chain. So their importance cannot be overstated. Up next is the American or Gulf butterfish, 14.4% of ocean eDNA. Again, another incredibly important forage fish. One of the interesting things about this, though, is we did not find them in our fall sampling bout. And that makes sense, though, because typically in the fall and winter, they're offshore, they're in deeper water. They only come they only come close to shore once that water starts to warm up. So it makes sense that they were completely absent, and then now here they are. They're a big, important player in our ecosystem. Makes sense. In the third position, we have Mumachug, 10.7%. This was somewhat surprising, not because they're here, but because I suppose because they're here in the marine environment. What I would imagine happened is that one of the stations near an estuarine site, say North Sandy Hook, say Shark River Inlet, or maybe Manasquan Inlet, probably one of these Mumachugs, one of these sites, we probably sampled it on an ebbing tide, and a lot of eDNA from the estuary was washed out, and so we have a big proportion of a highly important estuarine species. We touched on river heron, and so finally in our fifth position we have bluefish, 7.5%. Many of you guys grow up here, live here, anglers, incredibly important fish ecologically, as well as recreationally and culturally. They're ravenous predators. Really, really cool fish. We do have some honorable mentions here that, not part of the top 5, but still worth discussing. Right here, 3.4% is tautog. Interesting. The reason I bring them up is because if you'll recall from our fall sampling bout, the tog was at the #1 spot in the ocean. So it's interesting to see such a reduction. I'm going to have to think about this and get back to you guys as to why that may be. But interesting finding. Down here, dolphin, 3.8%. Obviously, a dolphin is not a fish, but because our Teleos primers pick them up so well, we like to get these in just to make sure that marine mammals are represented. And then finally, last ocean teleos I'll talk about is we have here the sandlands, 4.7%. Sandlands, they're an important forage fish as well. They have a kind of a cool behavior where when they're startled by a predator, they'll go and swim down to the sand and burrow into the sand, which is pretty cool. The interesting thing about them, though, I think from my perspective, is that the NOAA says that they have a hard time studying them because of how small they are and because of how difficult they are to catch in a traditional net. And so because they're difficult to capture in a net, they have a hard time studying it. But we have no problem finding them. So it kind of just goes to show some of the use cases of environmental DNA, which I think is pretty cool. Now we'll discuss the estuary and Telios communities. Mumachug, 27.7%. Again, as I was saying, an incredibly important estuarine fish. They're very resilient against changes in dissolved oxygen, in changes in temperature, in changes of salinity. And these factors make them prolific in the estuarine environment. They're mostly living by vegetation, by pieces of structure and kind of living like that. In the number 2 spot, we have the Atlantic silverside, 24.9%. A schooling forage fish, typically further out in the structure, also very resilient as estuarine fish go. They're kind of culturally, they're important because they're used as bait frequently. So if you see people throwing minnow traps into the water, they're after one of these two guys to fish with. In the 3rd position here, we have Atlantic or Gulf menhaden. As I said, incredibly important fish, really key to the continued functioning of the ecosystem. And then finally, for the estuarine Telios, we have the summer flounder, 5.9%. This is interesting because it's in a much higher percentage of our estuarine eDNA than last sampled out. As many of you guys know for surely, really big marine or estuarine predator. They'll eat basically any of these forage fish that care to have the misfortune of coming by. So now we'll move on and we'll look at the elasmobranch community composition. So here in the ocean, we have the smooth dogfish, 68.3%. Really, it's an opportunistic predator. Typically, they live a lot near the bottom. They eat things like mollusks and crabs, small fish, stuff like that. They're often caught as bycatch. You see a lot of them, so it makes sense that they're as prominent as they are. In the number 2 spot, we have clear nose skate, which is 18.4%. Kind of a similar case. As opposed to a smooth dogfish, which lives near the bottom, clear nose skate live on the bottom, eat similar foods, and also commonly caught as bycatch. Finally, in this 3rd position, we have winter skate or little skate. It represents 11.4% of the marine elasmobranch catch. The winter skate grow fairly large, actually. They can get up to 5 feet long. They have a similar diet as to the other elasmobranchs, mostly crabs, mollusks, bivalves, et cetera, small fish, stuff like that. The interesting thing about them is there's a commercial fishery for them, and there's they exhibit some pretty exaggerated onshore offshore migration seasonally, as many fish do. Now turning to the estuarine elasmobranch community, we have the same species basically in similar proportions with the addition of a cow-nosed ray, which is 1.8%. It's a schooling pelagic ray, meaning they swim up in the water column as opposed to sitting on the bottom like other skates and rays. They exhibit, if you're out on a boat, you'll often see them. They kind of exhibit this cool behavior where they'll jump out of the water and belly flop down, which is pretty cool to see. We were out helping our sister lab, the Dutton lab, and we caught one of them. So it was pretty cool to see one of them in real life. So in addition to what we have here represented in the actual donut, we also have some small other DNA that's represented, and I'd like to talk about them because they're pretty cool. So we caught some thresher shark, which was pretty cool. It's a large predatory shark. It typically targets schooling fish, and it uses this long tail as a whip, basically, to stun prey, which is pretty interesting to see. It's on Shark Week pretty frequently. Up next, we have the sand tiger, the medium-sized predatory shark. We caught this last year as well, way up here in this station, up at the mouth of the Raritan River. This year, it's not nearly as prolific. It's a medium-sized shark, typically eating things like fish, mostly fish, squids, crabs, etc., stuff like this. So one of the interesting things about sand tigers is they're not ram ventilators, meaning the expression is that sharks have to keep swimming. That's not necessarily true for these guys, is they can actually come and rest and sit still for a while, and they'll do that adjacent to structure during the day, and then once it's nighttime, they'll go start hunting again. And finally, we have the blacktip shark, medium-sized predatory shark. Typically, it's targeting like schooling fish. And when they're hunting, they can kind of exhibit a cool behavior where they jump out of the water in this acrobatic, twisting, jumping motion, which is pretty cool. So now we're kind of winding down. I just want to kind of summarize what we talked about. Is we found 79 different fish taxa, and we successfully sampled roughly 81% of the total diet richness in our region. We found that there wasn't a difference in estuarine and marine richness, but we saw that the community compositions did differ, and they were, while they were the same number or similar numbers of species, the species that were present were quite different. And I'd like to share a quote with you guys, which is that which is measured improves, and that which is measured and reported improves exponentially. That's from Carl Pearson, the father of modern statistics. I wish I could promise you that just doing what we're doing could help the environment and could bring about exponential increases in environmental health. I can't say that, but what I can say is that what we're doing is we're monitoring the environment, and we can advocate for policies that better the environment. And by doing that, we can make a real meaningful difference to further the cause of biodiversity. So in conclusion, with the help of you guys, we sampled over a geographically wide, oceanographically complex ecosystem, and we did so effectively. And hopefully we communicated cutting edge scientific techniques and kind of shared the importance of biodiversity with a group of you guys, a empowered group of community scientists. So we're doing this again in October of 2026, and I hope you guys are interested in participating. So thank you very much. So I'll take any questions if you have them. Yes.
Does vary by depth of the water that's additional or how you go?
Yeah, so I mean, what we're going to be seeing in a case like that is it will vary. Is where basically what's happening is if we're sampling in the Shrewsbury or Navesink River, we're sampling a very different ecosystem than way off here in on the shelf. It's an entirely different ecosystem, and depth is one of the most important contributing factors for deciding not only how many species are present, but also what species are actually there. So yeah, that's a very important.
Is there any way to fish at certain times in the year, or you have to have constant sampling?
We'd have to have pretty routine sampling to do something like that. That's part of one of the trouble with eDNA is that we'd have to actually get out there and physically sample it. We do have we're putting together a time series. One of the other parts of my job and some of the other lab work is that here in Monmouth Beach, I am out on the water every 2 weeks, taking water samples, and we're hoping to get a really interesting time series out of that. So with something like that, we will see, at least at a much tighter scale than every 6 months like we have here, we'll get a time series with a resolution of every 2 weeks. And we will start to see these seasonal changes happen at a much higher resolution.
Are you taking samples further away from the shoreline, or are you doing like what we did here, basically taking 2 steps off?
We're doing shoreline sampling for that one as well, so. And that's okay because eDNA does have a, like Dr. Adolph said, is eDNA has a half-life of about a day. So you can imagine that if I took a float and put it in the water and let it float, float away, is in one day, it could get pretty far. So yes, we're sampling right here, but we're still getting a fairly large area. And precisely how what I just described works is still kind of up in the air. I feel like I feel like eDNA sampling is a more localized phenomenon than some other people feel, but the science is we're still, in the process of discovering that. So, yes.
I remember last presentation, you were talking about some fish that were tropical that were found in this area. Did anything come up this presentation?
We found no Florida pompano in this season. Probably what happened is we sampled in like October of last year. What probably happens is that we kind of accumulate these tropical migrants, and they'll live in our estuaries and in our ocean, basically while the water's still warm. And as it starts to cool down, they probably die. And then we stop picking them up. So what I imagine is that if you get washed up into the Gulf Stream, you just don't live long enough. The water's still too cold to, kind of get and try and make your way into this ecosystem. Is what I would guess.
That the cow nose ray that Rich showed, that was found from y'all data set here. There's a Brazilian, I don't know if that was the Brazilian cow nose ray. The right one, but there's a Brazilian cow nose ray that some years back, our colleague Mark Stokel from Northstar found in eDNA. And, we're basically, I think it was he was the first person to show that a Brazilian ray species migrates all the way up here from South America. And
Amazing.
yeah,
Amazing.
A lot of our data.
Wow,
yeah, we do find it other times.
like the Monmouth butterflies. Yeah, amazing.
Yeah. So we don't have any humpback whales?
No whales. Well, no, we had whales, but they were tucked away in the
Others,
In the others,
yeah, they were tucked away in the others. Yes.
this may tie into your time lapse that you're going to look at, but the thing that's sort of gnawing at me, I think I asked you on the phone, looking at tidal stage, because the remnants, the residual DNAs, it's floating with the tide, so I would love to see if funding was infinite, Valley G, does the density or if you quantify it over different tidal stages. You made a comment about Mr. Fundulus, I guess, the fact that he was such a dominant guy in one of the stations might have had to do because it was so close,
Yeah.
To the inlet there.
Yeah.
I'm just feeling like, on an ebbing tide, if there's stuff floating in the water, it's I'm just stuck.
Yeah. So particle transport is, I kind of alluded to the fact that one of the big open questions in our field is how far does eDNA travel and how, what resolution can we actually sample with? One school of thought for how we can resolve this is to model, with a computer and do particle transport. And it kind of becomes a physical oceanography problem. And so that, that's an important question. And yeah, it's a lot of people are very interested in precisely that. Yes.
We recently had in Union Beach, a scandal about the pollution in that particular area. Does the species of fish change in relation to, lead in the water or, toxic metals?
That's an interesting question. I think that it would, where's Union Beach? Is it Irish?
Probably more around 42.
More closer to 42, okay. So, yeah, as we pollute the environment, we are gonna kind of start to knock out some animals. Some animals need it to be really quite nice. Other animals like mama chugger are much more resilient, and it takes a lot to kill a population of mama chug. So probably as an area gets more and more polluted, is you do start to lose species, and probably as you pollute, you're left with just the most resilient, and that's largely it. I was working here as an undergrad, as a deckhand, and we were doing some contract work for the NOAA. We were working up in the Passaic or the Hackensack, somewhere way up here in the Gateway region of New Jersey. We were actually trawling for black sea bass right next to the Superfund site that used to be where they made Agent Orange. And so it was, yeah, not great, not high quality water. It was not a beautiful day on the water, but we were sampling, and we were just coming up with nothing. And so that's probably what we're seeing, is just the water so polluted and so nasty that there's just nothing can make a living there.
You could, put a DNA, do the metals show up?
No.
And
No, so they don't show up in the sample because it's perfect. I have a lot of slides to get through, but I have a good. Oh. So this is basically the extraction process. This is what you guys did. You went down and you collected the eDNA. You handed it off to me, I filtered it, and then this is kind of the whole extraction process. And so what's going on is we're basically taking cells and breaking open cells to get access to that DNA. And then we're sticking them onto these silica filters because the DNA has a very strong affinity for silica. And then we're washing them off with an elution buffer. So the same water, basically what I'm saying is that this process, the extraction process, it's purely a molecular process. But if we gave you guys a second water bottle and we sent that out to some other lab, or maybe we could do it as well, we could test for zinc, we could test for aluminum, other heavy metals that may be disadvantageous to human or fish health. So, but no, we don't pick up metals in our eDNA process.
You asked that question because of the recent stories coming out of that coastline, Petersburg events.
No, I haven't had a general understanding. The oceans, words, so.
Because it's just my opinion. In order to see if whatever is coming out of there, or what they're testing for now, they've really figured, you'd have to have started testing 20 years ago for water, for the species, because now it's discerned, it's too late. Whatever is in there, that the fluid that is kills off what it's probably going to fill off at this stage of the game. So you would have had to start this. However, with what you're doing, a few years down the road, doing this twice a year, times a year, you might discover something more like that.
I'm also wondering, because it affects the animal fish,
Right,
would it be similar?
right. Because some of the super fun sites like, well, I'm get rid. But one of the prison waters, there's still benzene coming out into the river, right? So that.
Yeah, you're right.
You don't sample for this that far up, but if you sample in Boundbrook or Edison over time.
The baseline sampling, nobody's gonna fund you to sample for 10 years because you think there might be a problem 10 years down the
Years from
line.
now.
They need baseline data for it. Very difficult to get prolonged monitoring programs, get that sort of baseline. For offshore wind monitoring, we had 2 or 3 years of baseline sampling before they're gonna start construction, but then it never started construction, so we only have baseline sampling. That's our, I believe there was a question on Zoom.
Yeah, you're on Zoom. Can you unmute and ask your question?
Can you hear me?
Yes, yeah, we can hear you.
I was just wondering if larger fish leave more DNA behind than smaller fish. And how does that interfere with some idea of counts of how many fish?
So that's an excellent question. Larger fish as like when we're looking at like absolute volume of DNA shed, larger fish do shed more DNA, but it's more complicated than that. Probably the largest fish we have, let me minimize this, okay? Did I get, okay. Probably the largest fish we have is the thresher shark. I don't know, I can't get rid of that. Probably the largest fish we had was the thresher shark. But it's important to note that size is not the only factor that impacts eDNA shed. Things like temperature, things like, each species may have different tendencies to shed things, like a bluefish, for example, may shed more DNA than a dogfish just because of it's a ray fin fish. It has scales to lose. It has slime, et cetera, et cetera. A shark doesn't have that. It's kind of a much more it's kind of a much more sleek animal. So that's the case. But also other factors like say temperature and stress, and there's all types of stuff. And what time of year it is, if they're breeding, for example, many fish just kind of put gametes into the water. So there's a lot at play there. Do you have anything further?
Everything you said, plus think about a 500-pound tuna versus 500 pounds of menhaden, each of which weigh maybe 3 pounds. The menhaden, because they're smaller, lots of small bodies, is going to give up more DNA for that 500 pounds of fish than the 500-pound tuna will. And I believe it was Judith who asked that question. That's one of the big questions that we're all trying to figure out in understanding what the eDNA tells us about fish. And one of the ways that we do that in our lab is like the New Jersey Ocean Trawl Survey, they go out and they catch fish in a net, pull it up and count them and weigh them and everything. We have a bottle of water, and we do the DNA, and we're able to compare our results to their results to get at those things. Like if they catch mostly striped bass, do we also see mostly striped bass in the DNA or not? Or are there differences? Turns out sharks don't shed a lot of DNA in general. And menhaden do, and slimy fish do. That's what I would add. Thank you.
So is it possible to weight the eDNA results with the catch results?
Yes. Yes, and in terms of relative abundance, how enriched or not enriched a fish is within the trawl versus within the eDNA, we get a good concordance between the two. We tend to see the most DNA of the taxa, that they also see the most DNA in the net. What we're working on, and it's a subtle difference, but very important, is getting the absolute abundance. So when you do a fish stock survey, you want to know per hectare of ocean surface, how much tuna is down there. That's very difficult for you to do. But we and other labs are getting there. That's what everyone wants to. The federal government, in terms of fisheries regulation and management, knowing what the community is composed of and how many taxes there are in biodiversity is great, but they want to know how much fish are in the ocean, pounds, bushels, how much can we take? That's the big question.
So in the beginning slide, you said if you get 47 participants, you'll be able to get a 97% result of species in this area?
So that's what this is the thing with species accumulation curves, is it's kind of an idealistic situation. It's kind of an idealistic situation. So if we have 47 more people sampling, if we triple our sampling effort and it continues to follow this line, then we should find 95%. We should find 95% of 89.13. It's a species accumulation is you're doing math to kind of make statements about the environment. The trouble is that as we take eDNA samples, and one of the things that eDNA is very good at is that we continue to sample and we continue to find more rare fish. So if we take 47 more samples, that's great, but we're going to keep finding rarer and rarer fish. And as a result, we're basically species richness is going to continue to go up, which means it's increasingly difficult to get to the asymptote, and it's increasingly difficult to get to maximum richness. So if we say we used our entire data set to make this, so all of the fish, every animal, no matter how rare they were. If we used only fish and other species that contribute more than 1%, we would have already hit saturation. We already would have accounted for all of the fish that is in the environment in a proportion that's higher than 1%. But as we continue to sample, we're going to find them. We're going to find the important species, but we're also going to find more and more rare individuals, and that's going to change the map.
And can you go back to the other number, the 400,000 to 700,000? Can you explain that a little bit? Is that the?
Yeah, so there's a little bit of an asterisk here, is because this is all eukaryotes. So that includes, this number includes fish, but it also includes things like polychaete worms and corals and sponges and sea anemone. It's everything. So this is we've found 220,000 marine eukaryotes, which is a type of organism. We estimate that there are probably these many left, but we know for a fact that there are 16,475 marine fishes. We have described those. We know that to be true.
So that's almost 1 million marine species that are unclassified.
Yeah, that have yet to be described. But the important thing to know is that the vast majority of them are like things like flatworms, things that you could never see, you could hardly see with the naked eye, and things that you'd need to do genetics to identify as a new distinct species.
But they're part of the ecosystem though, right?
But they are part of the ecosystem, exactly. So we're interested in sampling these environments. We're just concerned in these samples. We're just concerned with fish. We sample with teleost primers and elasmobranch primers. But we want eventually to, and thanks to Grace is going to be helping us with that, we're going to be sampling with primers targeting eukaryotes and bacteria. So when we do that, we're going to see that total diversity. All life is, everything basically, with an asterisk, but.
And the bacteria be able to unleash or identify viruses?
It's an excellent question. I don't
Yeah,
believe
bacteria.
I don't believe viruses would, because they work with RNA, don't they?
Tough for me.
Yeah, we probably wouldn't capture them, and the size of the filter that we use, the holes in the filter are like half a micron in size, and viruses will go right through that. So we don't have an efficient way of capturing viruses.
That would prevent some of the dolphin kills or the seabird kills this past winter. And
Yeah.
Wow.
The menhaden fish kills a couple years ago.
Yeah.
No bacteria.
Yes, Tom.
How did you, derive the estimated total diversity number? Many physical catches or
Where did
the 87 to 101 to?
did that go? Is that down here?
You're supposed to-
Oh, I have,
Yeah.
oh, no.
No, it's in your results.
Yeah, so, we use a computer programming software, I suppose. It's called R Studio. In R Studio, we're able to use just a particular piece of software that is, if we feed it ecological data, it can tell us a lot. And it's kind of one of the uses for it is to calculate these numbers in particular. So that's how we got that. So that's one way to derive estimates of total diversity or total richness, I should say, is to use that type of software. But we can also use these species accumulation curves, and this asymptote, once that line levels off, that's another way to think, okay, this could be an estimate of species abundance as well.
I think I have a good way to explain that. It's called a rarefaction curve. So suppose, Nick goes out and samples fish, one sample. He finds bass, tuna, and a salmon. Then I go out and sample, and I find a bass, a tuna, and a, black sea bass. So combined, we each found 3, but our 2 samples combined, we found 4. Now, Heather goes out. She finds black sea bass, tuna, salmon, and a thresher shark. I think there was a 5th one in there. So now we've each found 3 taxa, but our 3 combined samples adds up to 5 unique taxa. What that's showing is that happening with the samples, and you can compute out to where's the point where if another person takes another sample, they won't find anything new. They may only have found, 5 things, but there's nothing new being found anymore. The accumulated taxa found in like 20 or 25 samples or whatever can equal 30 or 40 or some high number. But we both each only found 5 things, and nobody's finding anything new anymore. That's how you know you've saturated, you've reached the maximum richness. And that graph is a way of estimating that from samples that you have. The red part is our sample, we got that high, the black dot, that dotted line is extrapolating that out.
Don't actually know what some of those are
That's right, so that's a mathematical way of estimating
based on the environment and what's.
based on the sampling that we've done, estimating how much, how many taxa there would be if we kept sampling. And like Rich said, with eDNA, the problem is you keep finding all sorts of weird, really low level stuff in eDNA that, is like 0.01% abundant, and so it never really flattens out. You always find something new with every sample.
So kind of the other way is our best estimate, as I said, 97. We believe this is our best estimate because other ecologists like to use this method of estimating total diversity, but it's our cow estimate. And how that's calculated is we basically have the number of species that we saw. So in our case, 79 plus the number of, they call it singletons, which I think is a funny term. It's the number of organisms that you found exactly once. So because thresher sharks were so rare and so, not very abundant, I think it's fairly safe to say that a thresher shark was a singleton. So you basically count up how many different organisms you found, and then you square it. And then you divide that by 2 times the number of doubletons. What a doubleton is the number of species that you've only found in 2 stations. And it's that relationship to, how many species have I found once? How many species have I found none? And that's kind of a way to get at that very same question. Yes.
If a shark, hypothetically, ate a bunch of salmon
Yes.
and then swam into an estuary,
Would you be able to detect the presence of sound?
No, that's a good question.
Yeah, of course.
And how great would your funding become?
So, one of the mantras we live by is that fish genes are not fish. So we find fish genes. Most of the time, they represent the fish population, but not always. So Atlantic salmon actually does not exist in our waters. In the New York Harbor area, but we find Atlantic salmon genes all the time. I think it's actually up there.
It's right here,
So
2.3%.
that 2.3% Atlantic salmon genes that we find in the ocean is runoff and wastewater. It's the Atlantic salmon you buy at Costco or the fish market or the locks, whatever. The farm raised salmon, that's all Atlantic salmon. That species does not exist as a viable population in our waters. Unfortunately, used to. And in Maine, there's still populations. But we find it. We also find bronzino in the water. European sea bass, bronzino, very popular restaurant food. When we sample around Manhattan, Hudson River, East River, we get significant proportions, still in the single digit percentages of bronzino genes. Bronzino is European sea bass. It does not exist in New York Harbor, the Mid-Atlantic Bight area where we are. We're finding remnant traces of fish genes from. You ever see a Manhattan trash truck squeeze the juice out of the trash it picks up? And that flows down the street in the storm drainage to the harbor?
That's it.
There it is. Yeah.
How does that interact with the half-life of the eDNA? Because that fish would have been, I mean, I said, the fish would have been dead a long time ago, but then you're getting it in the same.
Yeah, they're never really strong signals in the data, but it's there.
Okay.
And as eDNA fisheries people, we do have to think differently about what we find. Again, fish genes are not fish.
And the half-life we use for the 24 hours, it's marine eDNA, so it's not in the water. It doesn't have that half-life, like the same half-life. It would be different if it's like outside, like on the person.
Oh, okay, I see.
Once it hits the
Environment.
water, yeah.
Yes.
Would your species richness even out if your free public labor sampled during different times?
What do you mean by different tides?
Flooding tide, ebbing tide.
Okay.
Twice a day.
I see what you're saying. I would imagine, so there's gonna be a little bit of exchange on a flood tide. Sites here-ish are they are estuarine sites. They're gonna look more like ocean communities. That's true because one, eDNA moves with water as we model eDNA just with, particle transport. DNA itself doesn't move, it just is washed away with the current. But the other interesting thing though, is that fish do move. And studies out of Australia in semi-enclosed estuaries show that fish routinely make tidal migrations into and out of estuaries to utilize them as refuge. So yeah, tide would impact it. And there's definitely a gray area in which everything in ecology is really a gray area. It's all a big gradient, but there's a gray area around here-ish in which if I'm in an estuary is up for it's a bit open question. There's one way of thinking about it. There's a couple of different ways to classify salinity. When you think about salinity from a biological perspective, actually all of this is marine water. And only once we get about here-ish, in my experience, does it switch over into only the saltiest of estuarine environments. So also, after this big rain that we had, there's so much fresh water coming out here might be a marine environment. That very thing has been described by a marine biologist named Ken Abel out of Rutgers. So what an estuary is and where estuarine habitat begins and ends and et cetera, et cetera. It's kind of a more complicated question, but as I was saying, I believe that eDNA is largely a more local phenomena. So probably we still have estuarine communities, and richness is indicative of that. Yes.
You don't have enough to put a community scientist out of business, because that question prompted me, I mean, given funding, would you not be able to potentially automate the process of collecting the water with a buoy or something, and you could do 24/7 capture, process, and all.
Well, we're very excited about that prospect. Dr. F, do you want to speak more on that?
So in our lab, we do these community science projects. We always pair them with what we refer to as laboratory sampling. Actually, this is the other one that isn't really. But Aaron here has worked on the Coastal Lakes project, where there's community scientists collecting water chemistry, and then Aaron collects water chemistry for like the last 6 years. And what we look at is how the community data matches or doesn't match the laboratory data, because there's this thing in science like, oh, you can't collect good data, you're just people. I don't believe that. And actually, our data doesn't show that either. It shows the data are good. So we do lab sampling of eDNA on automation. There are so currently Rich goes out on a boat occasionally and collects stuff offshore out to the shelf break out by the estuary. And Aaron and Liz, who's left, Grace goes out on the boat too, go out and spend like 4 days on the continental shelf, ping ponging among GPS coordinates, collecting water samples. Yeah, we need a drone or a robot or something to be able to do that, because as much as Aaron loves spending 4 days in Atlantic City, bouncing around on a boat. But so the problem is those things are about $5 million as they make. So it costs us, I don't know, I think $14,000 for a seasonal boat trip. Pay for the boat and the rooms and stuff. So. Sorry. But like a drone, like a water collecting drone, you just fly out and. But that is the future. I hope to live to see it.
Did I see a question back there?
Just hypothetical, everything being equal, with some DNA, if you took like 2 drops of blood from different fishes, all things being equal, similar fish, similar living area, just one would test it one, and then 2 days later, test the second drop of blood for each fish. Would one might degrade a little quicker than the other? Could you account for that? All things being equal, I mean,
So
same drop of blood, same exact milliliter, whatever. It's typical, almost same fish.
Yeah.
Could one degrade faster than the other? Guess theoretically it could, but.
It could. I mean,
And could you account for
given
that?
the same exact conditions, it should degrade at the same rate. But I know you have an answer to this too, I think. But I'll just say, the fact that eDNA degrades so quickly in the environment is actually one of the greatest things about it for what we do. Because the local source of the fish where you're sampling is always going to be a stronger signal than the DNA that's washed in there from the estuary or downstream, and it's taken 2 days to get there. So that's going to have gone through 2 half-lives. It's going to be, that means there's 25% of it left, compared to the fish population that's local to the area that you're sampling at the time. That's always going to be the stronger signal. So if DNA didn't break down, we'd be getting DNA from tens of kilometers around us, and we wouldn't know where the heck it was coming from. But because it does break down quickly, the stuff that we measure represents the local community primarily, with trace amounts of distal, far away stuff coming in. that we can kind of make a judgment call that it's probably not the dominant thing there. You know, the top 10 species that are given sight are usually the local stuff, and then after that it's Bronzino.
I mean, I was basically gonna say the same thing. I like to think about it like this is, if you're at, say, like a music festival, right? There's a bunch of speakers playing different songs and they're all super loud. You're gonna hear one standing next to the speaker. You're gonna hear song A much clearer than if I went away and now I'm standing next to speaker B playing song B. And that's kind of what we're looking at is, yeah, is.
Really good analogy.
Right? So, and we're lucky, we're lucky that, there's, you know, the half-life is so limited because makes it more of a local phenomenon. We can be more confident that what we have here was actually here. So. Yes.
Next question, I don't know, so originally when they did DNA testing, which is probably back in the 40s and 50s, I'm trying to remember when, and they did it with live fish. This is where you got all your information from now? Is this how we did this?
No,
But when you're comparing to now, though, you're saying you already was,
the reference sequences.
yes, was done years ago, and it was done with live fish, yes? And
Yes,
my question
yes.
is back then, because I know when I was a kid, was tons more fish when we fished. And so they must have had more fish back then, and that are DNA registered, that you're trying to catch now, you're trying to find now, you'll never find those fish again. Because they're gone, I mean, right, is that?
So,
Yeah, go on, no.
I mean, so you're right. Animals are moving. There should, you know, there's a lot of change going on
Evolution.
right now. From an evolutionary perspective, let's say the first guy to see a mummichug, like took the thing, put it in alcohol, and then some guy later, once we had DNA testing, took it out of the ethanol, took a fin clipping, sequenced its DNA. Like this was the first mummichug ever seen by a Western scientist. Like a straight line right to DNA testing. Obviously, this didn't happen. But even if that was the case, enough time has not passed for there to be meaningful differences in the genes that we're looking at. Because the genes that we're looking at are buried so deep into the genome that there just couldn't be any difference in.
But how about if you're gone though? I mean, that's the Kelly, leaves that Kelly on.
Oh, I see what you're
All the
saying.
time, but you know, but blowfish, I mean, the blowfish are not, I mean, I see it once in a while, stargazers, kingfish.
Yeah,
I caught them like there was no other, back when I was growing up.
yeah, I mean,
And they must have been DNA, and now I'm looking, you know, are they coming up or they're not gonna be? You know, you won't see them, you're trying to chase them or find them, and they don't exist anymore, right?
Yeah, so there are historical eDNA data sets, there are historical capture data sets.
Yes,
That we can compare the eDNA data sets to, see if there's a difference in things like that. We do catch kingfish at the beaches.
okay,
The only place I've ever seen kingfish is in the surf zone and the beaches, which makes sense. Southern kingfish and
and do some of the fish evolve into different species?
the timescales over which we've been doing marine science aren't really long enough for that to happen.
Okay,
The timescale it takes for the new species of fish to evolve. I can put a number to it, but I don't suspect that anything that we've got a DNA sequence for has evolved with something else within the time period that we've had that sequence. The structure of DNA was just discovered in the 40s.
40.
Yeah. Watson and Crick.
Watson and Crick, there you go, yep.
And getting the name of the X-ray crystallographer, who was part of that discovery. But yeah, from the 40s to the 2026, that's not very long in evolutionary.
Sure.
Rosalind Franklin. Oh,
hold on. Hi, Judith, can you unmute and ask your question?
Do I have to?
Can you hear me?
Yes, yeah, we can hear you.
If a fish was in the estuary and ate another fish and then went out to the ocean and defecated, what you would think that the prey fish was out in the ocean, right?
So yeah, we kind of had a similar question before, and we do see that DNA can be moved around in some strange circumstances. We discussed things like Atlantic salmon in wastewater that kind of flows out of the kind of flows into the environment. Presumably, we also have a lot of pork and beef, stuff like that, domestic animals appear in our water samples as well. I imagine that there still would be enough genetic material in a fecal sample to pick up on that, what the animal is eating. Actually, no, I do know that it's true, yeah. So theoretically, that could be a way that eDNA moves around. So good question.
We have one more question.
Yes, briefly, could you, for fish that we've been pickling over the years and throwing them in formalin and isopropyl?
Yeah.
Would that impact the DNA?
Some fixatives and preservatives do more than others, but in general, we can get DNA from archive samples. Yeah, people do that. So I want to thank Rich for the presentation. And thank you all. I hope you do decide to participate in the October Bio Blitz. I think that's what we're going now, right?
That's what we're going with, yeah.
Yeah, just so much thanks. It's so awesome to see people coming out to listen to this kind of stuff, and the fact that you participated in sampling, and tell your friends. We need 43 more samplers. Shit like that.